Hello everyone, I am Trexie Dano, one of the Group 9 member, and our topic is all about aspects of gene therapy. So, medical science has detected many human diseases related to defective genes. This type, this type of diseases are not curable by traditional methods like taking readily available medicines. Gene therapy is a potential method to either treat or cure genetic-related human illnesses. In 2015, a team of researchers at the Harvard Medical School at the Boston Children's Hospital stated that they were able to restore basic hearing in genetically deaf mice using gene therapy. So the Boston Health Children's Hospital research team also reported that they have restored a higher level of hearing down to 25 decibels, which is actually equivalent to a whisper. So human gene therapy was actually first realized in 1971 when the first recombinant DNA experiments were planned. It can be simply viewed as an insertion foreign DNA into a patient's tissue that hope to um, successfully eradicate the target disease. It was actually inspired by the success of recombinant DNA technology which occurred over the last 20 years. Without a doubt, gene therapy is the most promising yet possibly unfavorable medical field being studied. So the basic process. There are several approaches to gene therapy. These are the following: replacement of mutated gene that cause disease with a healthy copy of the gene, inactivation of a mutated gene that is functioning improperly, introducing a new gene into the body to help fight a disease. In general, a gene cannot be directly inserted into a human gene or cell. A gene is inserted into another gene using a carrier or vector. At present, the most common type of vectors are viruses that have been genetically changed to carry normal human DNA. Viruses have evolved a way of encapsulating and transporting their genes to human cells in a pathogenic manner.
Two types of gene therapy. The idea of gene therapy is based on correcting a disease at its root, fixing the abnormal genes that appear to lead to certain diseases. There are essentially two forms of gene therapy. So first is somatic gene therapy. So what is somatic gene therapy? So somatic gene therapy involves altering the genes in the body's somatic or non-productive cells. These changes affect only the individual patient and are not passed on to their children. So how it works? So a corrected or new gene is inserted into the cells of a specific tissue or organ like the liver, lungs, or muscle to treat a disease. One of the therapy is done, the changes stay with the individual. The other form of gene therapy is called germline therapy, which involves the genetic modification of germ cells or the origin cells that will pass the genes on to the next generation. So germline therapy involves altering the genes in reproductive cells, sperm, eggs, and these changes are inherited by future generations.
So my topic is stem cell gene therapy. Stem cells are mother cells that have the potential to become any type of cell in the body. One of the main characteristics of stem cells is their ability to self-renew or multiply while maintaining the potential to develop into other types of cells. Stem cells can become cells of the blood, heart, bones, skin, muscles, brain, among others. There are different sources of stem cells, but all the types of stem cells have the same capacity to develop into multiple types of cells. So, stem cell gene therapy combines stem cell technology with genetic therapy to treat disease, particularly genetic ones. It involves genetically modifying stem cells to express the therapeutic genes that can correct or replace faulty genes, offer regenerative potential, and modulate the immune system. Here is a more detailed explanation. Number one is stem cell basics. Stem cells are the body's building blocks capable of self-renewal and differentiation into various cell types. Number two is gene therapy basics. Gene therapy aims to treat diseases by introducing, modifying, or deleting genes within patient cells. Number three is combining stem cell and gene therapy. In stem cell gene therapy, stem cells are genetically modified into allowed to express therapeutic genes. Number four is therapeutic applications. Genetic disorders. Stem cell gene therapy is particularly promising for treating genetic diseases where a faulty gene is the cause. For example is sickle cell disease, beta thalassemia, and certain types of immune deficiencies are being explored with this approach. Number five is the advantages. Potential for cure, long-term benefits, and personalized medicine. Number six is risk. It is gene therapy can have side effects including immune reactions, graft versus host disease, and other applications. Number seven is the examples of stem cell gene therapy. Number one is CASGB, a gene therapy for sickle cell disease and beta thalassemia that uses stem cell to increase the production of fetal hemoglobin. So, a stem cell gene therapy developed by that uses mesenchymal stem cells to develop a functional collagen type 4 gene for treating epidermolysis bullosa. So that's all. Thank you.
To discuss of gene therapy. There are ethical issues involved in gene therapy. Some inquiries cite are number one. How can good and bad use of gene therapy be distinguished? Number two. Who decide which trait are normal and which constitute a disability or a disorder? Number three. Will the high cost of gene therapy make it available only to the wealthy? Number four. Could the widespread use of gene therapy make society less accepting of people who are different? Number five. Should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence, or athletic ability? Another controversy involves the germline therapy. As discussed, germline therapy is genetic modification of germ cells that will pass the change of the next generation. There are a lot of questions on the effects of the gene alter to unborn child next generation. Since the alteration can be passed on in the United States, the government does not fund researches on human germline gene therapy.